you. It's my pleasure to introduce Kate Carroll, who is scheduled actually to speak in my session on Thursday. We're fortunate that she was able to make it, and we fit her into this slot. A little bit about Kate, who's really done some cutting-edge work in chemical biology. She received her BS from Mills College in 1996, PhD from Stanford in 2003, and she did a postdoc in Carroll and Bertozzi's lab at the University of California, Berkeley. She's really done some outstanding work on redox chemistry related to cysteine and cell signaling. And the title of her talk is Cysteine-Mediated Redox Signaling, Chemical Tools for Biology.
Great, thank you. I think I've got a mic here. Okay, so I feel a little bit like a fish out of water here, but hopefully what the take-home message I think today will be that sulfur chemistry, whether or not it's at the cysteine level or methionine level, small molecule or peptide level, is quite complex, and that's what we're experts in. We're experts in sulfur biochemistry and chemistry, and we think a lot about that, and those chemistries can be leveraged to design some very unique molecules with unique properties, both for basic discovery, but also, we believe, for therapeutic purposes as well. So the overarching goal of work in my group is to really understand the role of redox systems and how they control very complex biological events vis-a-vis sulfur oxidation, with a particular emphasis on sulfur post-translational modifications, of which there are a variety, and I'll show you on the next slide. But in a nutshell, what we're concerned with is really there's a stimuli at the outside of the cell surface. This translates into oxidation reduction at the protein level. This changes protein behavior, peptide behavior, and ultimately transmits into changes in dynamic cellular responses. So this is the workflow that we're most interested in, developing reagents that allow us to really understand these reactions, monitor these reactions, and track them in cells to understand, again, the basic mechanism of action, but also to understand whether or not there are avenues for therapeutic intervention. And so really we always, although we're chemists, we start with the biological question or biological phenomenon that we're trying to understand. Oftentimes, in order to dissect this very complex behavior, it necessitates the development of chemical tools. Ultimately, we do a lot of systems biology in my group, and then at the end of the day, what I'm most interested in is molecular mechanism. That is, how do these changes at the atomic level then transmit into changes in structure and function? So this is a list of just a few of the biologically relevant sulfur chemotypes. So I'm showing you cysteine chemotypes, if you will, and everyone is very familiar with the thiol and also with the disulfide. But if you take a larger glance at this slide, you can see sulfur, which is present in many, many different forms. Perhaps if you were involved in cardiovascular research, you're already familiar with nitrosethiols. But really, the scope is quite large. And for those of you who think about disulfide formation in peptides and are concerned potentially with oxidation there, these reactions can be quite facile to form the thiosulfonate or the thiosulfone with subsequent concomitant disproportionation. So each of these modifications of cysteine represents a completely separate post-translational modification. So all these are small atomic changes. They dynamically change, dramatically change chemical reactivity, redox activity, and also most logically metal binding. You take a soft thiolate and convert it into a harder center, and you get gain or release of metal ions. And so what I want to point out here with this next sort of illustration is the fact that sulfur can exist in so many different redox states within biology. So I'm showing you 10 different states on this particular slide, including half or fractional oxidation states. And so the point again here is that there are many different biologically relevant cysteine derivatives, each with their own reactivity. And so from our perspective, from a chemist's perspective, or chemical biology perspective, really, we viewed this as an area really to develop methods around the detection, which will be key, of course, to fully molecularly describe these reactions and understand their role in biology. So I'm going to spend most of our time today, because we don't have that much time, but I'm going to talk mostly about sulfenylation. So SOH, that is the reaction of a thiolate, generally with hydrogen peroxide, but many other biological oxidants and other reactions can lead to sulfenic acid. I'll tell you about that in a few minutes. But just to give you sort of the bird's eye view of what sulfenylation can do, despite the fact that it's a very small modification, is that it can regulate stability. It can inactivate function, for example, if you require a catalytic cysteine residue. It alters protein-protein interactions, subcellular localization, and also metal binding properties. And the story that we're most well known for is that it can augment function, for example, in EGFR. Oxidation of a critical cysteine residue in the active site not only modulates the binding of activators or inhibitors, but also stimulates kinase activity. And so there's a lot that's going on here, a lot of potential biological function here that we'd like to understand. Now, it's not just two-electron oxidation that results in sulfenic acid chemistry or production at sites of cysteine. So this is the most traditional or most focused on canonical route, where you have hydrogen peroxide, which is produced biologically for signaling purposes, leading to sulfenic acid, or there can be alkyl hydroperoxides as well. But peroxynitrite and nitrous acid are also quick routes to sulfenic acid, very aggressive. So these are two-electron routes, and then also there's hydrolytic reactions. So for example, hypohalous acids can be hydrolyzed to sulfenic acids quite readily, and this is a big deal, as particularly in the immune system. That plays a very relevant role. And then also hydrolysis of nitrosethiols and also disulfides is quite common, and so that will also yield sulfenic acid. So there are many different biological routes to get to this particular intermediate, whether or not in a signaling context. Now, the status quo when we entered the field to figure out which cysteines undergo oxidation in a particular scenario is this very complex workflow of a sequential series of chemical reactions. So first you'd have oxidized and reduced cysteines. You'd use an acid trap to protonate all your thiols. Then you'd have to neutralize the pH, alkylate, block all your free thiols, and now you have effectively an oxy-ICAT or isotope ABP sort of workflow where you're looking at non-reactive cysteines versus reactive cysteines as a loss of signal. Now, a slightly more complicated workflow is to quench and remove the additional alkylating agent and then reduce that oxidized cysteine to free up a nascent thiol. Then you use another alkylating agent which is tagged in some way, and then now you can observe the appearance of signal. That is, that cysteine here was oxidized. So this is really the status quo. Now, there's a lot of issues with this particular workflow. So first, you can't look at all of the different oxidation states that I told you about earlier in the talk, all of these different oxidation states that have different properties. The acid trap destroys labile modifications. You have competitive reactions at neutral pH. These alkylators, shown here and here schematically, also react, it turns out, with oxidized cysteine residues, so then you're competing there. We're monitoring a signal loss. We're not really sure about how quantitative these chemical steps are and so on. So we really felt that there was a lot of room for improvement here with the notion of chemoselectivity. But I think the largest issue with thinking about cysteine oxidation or methionine oxidation at any level is to recognize that the cell is compartmentalized from a redox standpoint. So for example, the cytoplasm is generally quite reducing, as is the nucleus, whereas the ER and the endocytic pathway is quite oxidizing. So these present very different types of environments, which once you homogenize them, turn into a very oxidizing whole cell lysate. So I know a common workflow that people who are developing redox-oriented biomolecules or therapeutics, they'll look at glutathione and other types of thiol-based markers, and they'll always do it in this whole cell lysate, and they won't see any changes, and they'll say, well, our molecule or our peptide, our drug, has no effect on the redox behavior of the cell, but that's not true. They're just simply monitoring this state. So cells are redox compartmentalized, and that's really one of our claims to fame, is the development of tools that allow us to preserve that redox compartmentalization and probe these events directly in living systems. And so that's where chemoselectivity comes in. It's something that we think about a lot in our group, and it really is the mother of invention. So chemoselectivity here is where we have A reacting with B and nothing else. And so that's one type of chemical challenge. And then taking the next step forward, we want it to be bioorthogonal. That is, we want it to go in cells. We want it to go in the presence of water and nucleophiles and electrophiles and oxygen. So this is a tall challenge, especially when you're dealing with cells and you want to make sure that your molecules are selective and non-toxic. Nonetheless, what we can do is we think about this from a very basic organosulfur chemical standpoint. And we think about how sulfur behaves in different contexts. So in the context of a disulfide, unless you have a highly polarized environment, you have roughly equal sharing of electrons. And so there's not a lot of chemical activity going on with the disulfide. And the same is true of the thiol. There's slightly more attraction to the sulfur atom, but it's large, readily polarizable. Whereas with sulfenic acid, what we have is we have this electronegative oxygen atom just adjacent to the sulfur, and it's basically wicking off all of the electronegativity and the electrons. So we have a real unequal sharing of electrons here, and this is really the key to us thinking about how do we get reactions that go selectively with this type of functional group. And we can see that actually in electrostatic surface potential maps where the sulfur in a thiol is large, polarizable, nucleophilic, and the sulfur in a sulfenic acid becomes much more electrophilic. So we always think of sulfur as nature's nucleophile, but every time you add an oxygen atom, you essentially change the nucleophilic properties quite dramatically. And so it was in the late 40s actually that Foss discovered reactions of actually sulfenyl halides with nucleophiles, a simple displacement reaction, including carbon nucleophiles. And so it was in the early 70s actually that the parallel was made between sulfenyl halides and sulfenic acids, and that you could use carbon nucleophiles to effectively react selectively actually with sulfenic acids, resulting in a more or less stable thiol ether adduct. And you can control the stability of the ultimate adduct, obviously, through the differences in electron withdrawing groups and the kind of the functional groups you decorate your small molecule with. But the most, I think, well-known molecule that reacts with sulfenic acid that was discovered now many, many decades ago is called dimidone. It's a cyclic 1,3-beta diketone, which forms an enol in aqueous conditions. And ultimately, the mechanism is shown here, where we have a low pKa carbon, or the hydrogen is lost. The pKa is about 5 here. That results in a carbanion, and ultimately attack of the carbanion on the sulfur, leading to the thiol ether group here with concomitant loss of water. So now we've harnessed this particular reaction to develop a number of different chemical tools to study protein sulfenylation. So we've studied a lot in terms of trying to understand the mechanism, how we can control the reactivity of these molecules. We've developed dipeptide models of sulfenic acid. So for example, this is a cyclic sulfonamide that then opens in the presence of water to form a sulfenic acid. So we can store it on the shelf, but complete particular biochemical studies. We have tools for global analysis with chemical reporters like alkyne or azides, and we have tools for relative and absolute quantitation. So this is more at the proteomic level, which I'm going to tell you a little bit more about. We've developed an immunochemical approach that allows us to first diffuse in our small molecule probe of choice and then use an antibody that's been raised either globally against the chemical adduct or hapten, or against specific families or specific proteins. So for example, we have a tyrosine phosphatase antibody that works quite well that allows us to look at oxidation of this really incredibly important class of enzymes. Now we've also taken this carbon nucleophile warhead, if you will, and we've appended selective targeting molecules that allow it, both peptide and small molecule based, that allow us to target specific classes of proteins or enzymes, or specific proteins or enzymes in and of themselves. We have methods for site-specific incorporation, so we can incorporate this sulfone and with irradiation generate a sulfenic acid in situ. We have methods for solid phase incorporation of these different molecules, and we also have organelle-targeted molecules, and so this is an example of a mitochondrial targeted probe. So we've utilized all of these chemical tools at our disposal to really study this modification. And so one of the approaches that we've taken is very systems-based. And so this is kind of a grinded up, spitted out proteomic experiment where we have a control, either cell type, either organ, or in this particular case, cell line, and we have a stimulated group where we treat with hydrogen peroxide or we make a knockout, and so on. And so what this allows us to do is this workflow allows us to look site-specifically at what's happening at every single cysteine in a particular protein or peptide. And so in this particular case, we're looking at a molecule called FASN. It has a variety of different cysteines, and we can see there's over a 60-fold range of reactivity within this particular protein. So this really gives us a sense of how dramatically you can change cysteine reactivity within a single target. We can see this change even in cysteines that are adjacent to each other in three-dimensional space. So here's an example, peroxyredoxin 6, or FAB-P5, which has two cysteines which are just adjacent to each other. We can see this in the structure, and we have dramatically different sensitivity to hydrogen peroxide. And so multiple sulfenylated sites on the same protein can be very differentially affected by hydrogen peroxide or other types of redox reactions. And so prior to this work, we didn't really understand the full scope that was possible. Now, we've also mapped not just peroxide-sensitive cysteines, but really we wanted to take a step back and ask the question, when we have cells generate peroxide themselves, aka endogenous hydrogen peroxide production, what does that look like in terms of sulfenylation when we compared that to just adding hydrogen peroxide to cells? And so what we noticed is that certainly just the blanket addition of hydrogen peroxide without any spatial and temporal concerns resulted in a higher number of sulfenylated sites on more proteins. The increase or change in amount of oxidation in each of these sites was significantly higher. So we had more than a twofold increase on greater than 90% of the sites. So it's a way to cast a very large net, but perhaps not the most physiological pathway to take. And so when we treated cells with EGF to enable NOx enzymes to make endogenous hydrogen peroxide, we definitely identified sulfenylated sites, although there were far fewer, if you will, almost half, on 174 proteins, and a less increase on these particular sites. Now, if you look at the change in oxidation in EGF treatment versus peroxide treatment, of those that responded to both treatments, we can see actually quite good agreement. So like I said, peroxide is a way to cast a very large net. But what we also notice is that it's very indiscriminate. So we can see effectively no oxidation on EGF treatment of cofilin, whereas we see sort of this across the board oxidation on cysteines and cofilin with peroxide treatment. And so this is just another example then of the sensitivity and the difference between EGF and hydrogen peroxide. So the point here with this slide is that endogenous EGF-driven sulfenylation is considerably more selective and more specific. And it's not just EGF or growth factors that will generate the production of endogenous hydrogen peroxide. It's everything from growth factors to wounding to many different types of stimuli that will lead to changes in oxidation. So everything from cell death to cell proliferation. Now, we didn't just want to catalog or take an inventory of the key players. We also wanted to understand whether or not there was a functional significance of sulfenylation, right? We're just panning for cysteines that become oxidized. That doesn't tell us whether or not they are significant from a functional perspective. And so we looked at SIRT6, which kept popping up over and over again in our proteomic analyses. SIRT6 has a variety of cysteines. It was always cysteine 18, which is adjacent to the ligand binding domain, which became oxidized and highly oxidized in our studies. And so to make a long story short, we knew that SIRT6 functioned as a co-repressor of HIF-1 alpha, but we didn't really understand the molecular basis. And ultimately, what we found is that hydrogen peroxide oxidizes this critical cysteine 18, ultimately forming a disulfide-linked complex with HIF-1 alpha, and that blocks transcription from HIF-1 alpha start sites. And so that ultimately enables redox regulation of this entire paradigm here. So SIRT6 appears to function as a peroxide sensor. So again, when we see dramatic changes and it comes up over and over again in our screens, that begins to hint at functional significance, which is confirmed there. In terms of bioinformatic analysis, you might think that if we have indiscriminate oxidation going on at a cellular level, that we might see multiple sites of sulfenylation on any given target, but actually the majority of targets only had a single site, so almost 75% only had a single site of oxidation, and so multiple sites of oxidation were actually significantly more rare. We also thought at the outset that we would see a lot of buried cysteines becoming oxidized, but it turns out that exposed cysteines undergo oxidation significantly more actually than buried cysteines, and so we can see this. This is an example of a crystal structure where we have buried cysteines not undergoing oxidation, whereas we have surface exposed cysteines undergoing dramatic oxidation. Now, like other redox modifications, sulfenylation also has a consensus motif, and so the more sequences that we get, the better that we are at predicting sites of sulfenylation. Consensus motifs compared to nitrosylation or electrophile sensitive cysteines are CXXE, for example, so the presence of an acidic residue, or KXE. And we're not sure. In some cases, this is probably due to the depression of the cysteine pKa to allow the initial oxidation event, but also stabilization of the sulfenic acid modification itself. Now, a geoanalysis, this gets a little bit boring when you're talking about proteomics, but suffice to say that we found this modification throughout the cell, including places that are logical, like the mitochondria and other types of oxidizing environments. And we also found a real disproportionate amount of oxidation among key players in signal transduction. So kinases and phosphatases and ligases and deubiquitinases and acetylases and deacetylases. And so these initial findings suggested crosstalk between sulfenylation and other post-translational modifications. And so we've gone from a place of really not knowing whether or not these modifications form at all in cells to any kind of stable amount that we're able to detect, all the way from discovery in cells to inventorying to understanding their properties, to understanding their role in signaling, and ultimately in disease. So we've come a long way. And one of the paradigms, like I said earlier on in the talk, that we're most well known for really comes back to this notion of NOx control of EGFR activity. And so this is one of the earlier studies that we've done. And so what we've shown is that when you treat EGFR, when you stimulate, you activate the receptor with EGF. This, of course, results in receptor dimerization, but also colocalization and interaction with an enzyme called NOX2, which itself makes hydrogen peroxide, oxidizes an active site cysteine within the kinase domain, and ultimately elevates the overall level of tyrosine phosphorylation and signaling through this node. And so the sulfenylation occurs particularly on cysteine-797, but again, it activates the kinase. And so let's talk then about targeting that kinase and what oxidation of that cysteine residue actually does. So this is the kinase domain shown here. It has a variety of different cysteines, none of which are oxidized, with the exception of the cysteine in the active site, which is adjacent to the catalytic loop and the nucleotide binding site. So cysteine-797 is selectively oxidized. Now, of course, there's I guess we're revisiting this trend. It's becoming a lot more accepted now by big pharma. That is the idea of developing covalent inhibitors to target therapeutically important molecules. And so oftentimes these are targeted, most often are targeted towards cysteine residues in its reduced form. This is an example of 2-hirafatinib, which has made it through clinical trials for the treatment of lung cancer. And so what afatinib does is it has an acrylamide warhead that covalently modifies this particular cysteine within the kinase. Now, thiol-reactive covalent inhibitors are all along these lines, most generally actually this kind of a Michael addition type scenario where you have an acrylamide warhead reacting with the thiol to generate the corresponding adduct. Now, what happens, however, when you oxidize cysteine, whether or not you form a disulfide or not, even just a sulfenic acid, this completely blunts that chemical reactivity. So not only does thiol oxidation impact the regular function of these proteins or targets, it also impacts drug pharmacology. And so that's something that's really critical to think of, regardless of whether or not you're thinking about redox-oriented pathway or modification. You want to know whether or not your cysteine or your particular sulfur is redox active. And so we've shown this now looking at EGFR under chronic oxidative stress, a low level either hypoxia or simulated chronic oxidative stress. And we can see these two covalent inhibitors in the presence of oxidative stress, we have this refractory population of kinase that you can no longer inhibit covalently. And we can see this with both compounds here. So chronic oxidative stress can alter the pharmacology of these different inhibitors. And in cancer cells, the fraction of EGFR in this particular oxidized form, we've measured this now in tumors, and we've estimated it at 20%. It's actually a little bit higher. It depends on the patient, but it's somewhere between 20 and 40%. So it's not just some kind of boutique population. It's a very relevant population that we need to think about addressing. And what's more, it's an activated population of EGFR. So now we wanted to think about how we could exploit this, both for basic discovery, but with potential at least to develop molecules of more specific molecules, probes of oxidation of specific enzymes. And so what everyone is trying to do right now is utilize electrophiles, like I showed you on the last slide, and target semi-conserved cysteines in the particular target of choice. But the problem there is you have a lot of glutathione, you have a lot of different types of molecules that will cross-react with other cellular nucleophiles, whereas you have sulfenic acid, and we can exploit the fact that it's a soft electrophile, which is very rare in biological systems. And so what we do is we take this broad spectrum nucleophile and we tailor it, and you can tailor it with a small molecule or peptide, and ultimately develop a focused biological probe with quite good selectivity. And so this is effectively exploiting existing non-covalent inhibitors in conjunction with structure-guided design, that type of workflow. And so it's not like you just go to the Sigma catalog and you look up all types of electrophiles. We had to develop and synthesize, prepare, and discover these nucleophiles really from the ground up. And so we developed a library of over 150 both C-Nucleophiles and also linear nucleophiles that are both covalent irreversible and covalent reversible, depending on the application. And so these are able to target selectively cysteine sulfenic acid with rates of 100-fold less to 200-fold more, actually more than 1,000-fold more relative to that parent molecule that I told you about called dimidone. And so we've utilized this in a proof of concept experiment to go after the oxidized form of EGFR, simply taking this quinazoline scaffold and appending one of these less reactive nucleophiles. And we can see both with cells in culture, we can see redox-dependent inhibition of EGFR activity, and we can also see this in a tumor xenograft. And so again, just proof of concept, but the notion here is that you target this peroxide or redox-sensitive cysteine, which is predominantly in the oxidized form, or there's a good proportion of it in the disease state. And we've done this with tyrosine phosphatases as well. Now, this particular class of probes that we've developed, again, spans across a range of reactivity and also different chemotypes, if you will. And so we took for this particular manuscript 5 different molecules that reacted with quite different reactivity, but also had different 3-dimensional structures. This is present in the enol formation, these are in the keto formation, and had presented different geometric considerations. And so what we did is we screened, to avoid permeability changes, we just screened proteins generated under native conditions for their ability to react with these different probes. So we profiled to see whether or not we would see reactivity that only went with the chemical rate of reaction of these probes, or if we would see differences in reaction among these reagents. And what we found is indeed the most reactive probe did detect the most sulfenylation events, but then with the remaining 4 probes, we actually see quite dramatic differences that don't correlate necessarily to reactivity, but really correlate actually with the chemical structure of the probe. So we also saw a big difference in surface distribution analysis, so we're able to actually now access more buried residues as well. Now, we noted very diverse redoxome reactivity profiles. So this is a list of some of the proteins that we identified. This is a heat map. And we can see this is the most reactive probe, but then again, with the remaining 4 probes, we see actually quite different and quite privileged reactivity with certain classes of proteins. This is an analysis comparing adjacent sequences that the probes recognize. And we can see some sites, again, which are surface exposed, being recognized by the majority of the probes, whereas we have a very unreactive probe selectively recognizing and covalently modifying the sulfenic acid in CARM1. PTPs emerged as a privileged target of a probe that we call PYD. And we can see it fits very well in there. We were able to recapitulate these data, and we pulled out many, many different PTPs that reacted selectively with this particular probe. So we have next generation reagents that we're developing, both, again, covalent irreversible and covalent reversible, depending on your application. There's a variety of different chemotypes that we can use. It's not just the cyclic diketone. And we've also begun to identify hidden nucleophiles, both in natural products and also existing drugs. And so there's a lot of interest in profiling and really trying to understand what role these nucleophiles actually have in their mechanism of action. And so I'm going to close here, but I just want to remind everyone that it's not just sulfenic acid that we're interested in. We're ultimately organosulfur chemists, and we're interested in developing reactions that will allow us to detect all different types of sulfur chemotypes, and we have reactions, selective reactions for sulfenic acid, thiol-sulfonate, sulfite, persulfide, sulfonamide, and so on. And again, all of this chemistry exploits the differences in chemical reactivity for selective detection. And so just to wrap up, I've introduced 2 very simple new concepts that we are using, and indeed the field has moved forward to use to study redox signaling. One is this notion of developing selective chemical probes that allow us to detect distinct cysteine chemotypes. And 2 is to probe oxidation within a native cellular environment. That's really been a huge, huge hurdle that we've overcome. And the new chemistry and chemical tools have revealed, in this case, protein sulfenylation as a global signaling mechanism involving key regulatory players like kinases and phosphatases, acetylases, ubiquitinases, and so on. And then finally, I've introduced the concept of nucleophilic irreversible reversible covalent inhibitors, which represent an entirely new approach to pharmacologically profile and target redox-sensitive cysteines in therapeutically important proteins. So with that, I'll acknowledge the postdocs that have done the work, the alumni, my great collaborators, these agencies, and also pharmaceutical companies for their funding. Again, the organizers for the invitation, and I'd be happy to take any questions that you have. Thank you.